Hi, my name's Sophia, and this presentation is going over my work this summer in the Willett Lab at the University of Oregon Knight campus for accelerating scientific impact, and also mentored by Gabby Lindberg. I'm excited to share what I've been up to this summer and progress I've made towards my research goals. So, kind of starting back from the beginning and how I got where I am today. I started out in high school with many unexplained symptoms, and I was dismissed by many doctors, which led to my inevitable diagnosis of Ehlers-Danlos syndrome, which is a connective tissue disorder, when I turned 18 years old. This video of me is in high school when I was relearning how to walk and move and kind of operate with my new spine conditions after I had a concussion and had never fully recovered, which kind of inspired this passion in me to enter the medical field, what I originally thought as a doctor, but after getting exposed to research here at the University of Oregon, I had this passion in order to look into why these things were happening to me, and the scientific background and research beginnings began at that point, and also making women's health a priority. That's when I connected with my PI, Nick, and mentor, Cora Ferguson, who is overseen by both Nick Willett and Gabriella Lindberg, and kind of with our overlapping interests in kind of connective tissue health and EDS. So just some brief background on my condition. It's called hypermobile Ehlers-Danlos syndrome. There are about 14 subtypes of EDS, but the type I have is the only one without a known genetic marker, which makes diagnosis a little bit more challenging in some ways. It is diagnosed clinically using something called the Batten score, which looks at your joints and how hypermobile they are, and then in collaboration with other symptom criteria. Symptoms include joint hypermobility and instability, chronic pain and fatigue, and then multi-organ systemic issues. And this is often misdiagnosed and misunderstood, especially in women, which kind of drove my interest in female-specific research and looking into what differences may be in the prevalence of EDS between female assigned at birth versus male assigned at birth populations. So EDS, just kind of wrapping this part up, looks primarily like in the left video, relearning basic movements, recurring joint subluxations and dislocations. I'm a frequent flyer at physical therapy and have to manage my school and workload with that, and knowing my health is a little bit more dynamic than some of my other peers. I am a pro-surgical patient and know what it's like inside the operating room, but now I get to look at those tissues outside the operating room, which is really awesome, and I've learned to advocate for myself and my accommodations, which has also led me to some really cool opportunities because I feel as though I've gained those skills in order to share my story and advocate for change that I see in the world I want to live in. And on the right is just a picture of me, my first ever headshot, which is really fun, and it can also be fully invisible. The conditions I have aren't always super obvious from the outside, and I've really formed a strong sense of community with people who have similar conditions, and understanding the conditions I have and the pain I'm experiencing is pretty motivating. But kind of going into the next slides, some of the background I have motivating this work is EDS is far more prevalent in females, but another condition that also is osteoarthritis. So kind of looking into bridging science in my own experience, like I mentioned before, HEDS affects collagen, which leads to joint instability, chronic pain, and systemic symptoms, which is what inspired me to pursue this musculoskeletal tissue regeneration. But now I'm looking at why or how sex-specific factors and inflammation impact cartilage, which are key areas affected in EDS. So looking a little bit closer into cartilage, cartilage is a specialized connective tissue that provides cushioning and structural support to joints, and collagen is a key protein in connective tissue that provides structural support to these joints, so kind of acting as a bit of a buffer and cushion when someone walks or moves or runs, and over time it could be degraded, and collagen may look healthy and organized in it and have certain areas of different types, but other times when it's more of a inflammatory conditions, they might kind of degrade the healthy collagen and other collagen types that maybe are a little bit more fibrotic, kind of make the collagen and overall connective tissue more susceptible to degeneration and not holding that very cushion joint surface that we would like to see. So, as I mentioned before, cartilage is a very specific type of connective tissue that I'm interested in looking at. But in HEDS, connective tissues abnormalities may affect cartilage integrity and joint function in addition to other aspects. So I'm kind of narrowing on the specific area for right now and looking into how inflammation and sex-based factors influence the cartilage matrix. And as a patient scientist, I really am trying to bring lived experience into the lab, bridging my empathy for the work being done and innovation, and trying to kind of curate new ideas from my own lived experiences in collaboration with what I've learned so far. And then I just think overall including patients in research also fosters a huge aspect of relevancy, addressing things that do really matter in the populations that are affected, and urgency as well as deeper understanding. So in my research this summer, after all that background, I think it's helpful to kind of look at the different aims I'm kind of focused on. The first was identifying sex-specific disparities in literature within different populations, primarily female and male, assigned at birth groups. And here's some just quick stats I have from my literature review. But the first is rheumatoid arthritis impacts 3 females for every 1 male. The females have a 5 to 10 higher incidence of ACL tears, and 70% of those diagnosed with EDS are female, which are all different diseases, but the correlation with this higher impact and prevalence in females points to some sex-specific factors in disease pathology. So looking at this next slide, another interesting stat that really kind of sparked my interest in looking deeper into this research was between 1997 and 2000, 8 of every 10 drugs were withdrawn by the FDA for posing greater risks to women, and women still experience 50 to 75% more drug adverse reactions than men, which highlights the risk of male-centric research. And these trends have dire impact on the lives of biological females, not only with disease prevalence, but a lack of treatments, which is why we have an urgent need for preclinical models that will allow us to develop therapeutics in a female-specific context, which is missing right now. So knowing that, going forward into looking into one specific disease that predominantly affects postmenopausal females at a greater rate than males at that same age, as you can see in this graph on the top right. Approximately 10% of women and 7% of men ages 40 to 49 have knee osteoarthritis, as do 35% of women and 19% of men ages 60 to 69, which is a huge disproportionate impact, especially in those older postmenopausal populations. So knowing that, we look at the basics first, and knowing that osteoarthritis is a chronic degenerative disease of the joint. It has a huge annual impact on the economy in the U.S. alone, and it is characterized by chronic inflammation that causes cartilage matrix degeneration. So in my specific research, I'm looking at this summer, I established baseline controls for inflammatory conditions, looking at the control chondrocytes in a normal non-inflammatory media, and then adding in inflammatory signals into a different media condition where we'd essentially create these spheroids in order to expose these, essentially mimicking these cells in an environment that mimics the human body that has either healthy, normal conditions around it or inflammatory, angry, not super happy conditions around it, and looking at the differences in the cartilage matrix under each condition so that we can quantify it and use it as a baseline to compare further models along down the line too. So my second aim this summer was looking at sex-specific factors' influence on cartilage integrity in vitro. So kind of going along with the same experimental design, I was able to look at one specific sex factor and see how there were changes in the cartilage matrix pre- and post-inflammation, and that work continues to be ongoing. But I will hopefully have some results soon, and I'm excited to share at the Undergraduate Research Symposium in May. So look forward to hearing more about that in the future. And I want to thank you for your time and for this opportunity to present. I learned a lot throughout the summer and managing different work-life balances as both a patient and a researcher and a student, and being able to take on tasks like weekly lab meetings and manage my workflow and experimental design. So I really gained a lot from my work this summer, and I appreciate your time and attention to learning about my project. Yeah, thank you.